So in order of all of these events to happen and have muscle contraction, we need a connection between the nervous system and the muscle fibers. And in the nervous system, we have, if you can recall from chapter 4, when we were describing the muscle, the nervous tissue, we just kind of mentioned that the neurons, cells of the nervous system, has this long prolongation that is called the axon, and that can travel all the way, let's say, to the arm to reach the muscle here posterior in the posterior aspect of the arm, the triceps brachii. Now, in there, this is a bundle of those axons form a nerve, and the nerve go and supply the entire muscle, and the entire muscle um, gets penetrated by this nerve. The nerve branches following the layers of the epi, peri, and endomysium until it reaches right here an individual muscle fiber. So right in here we have an axon with and in its branches with one muscle fiber. Now, what happens in here? Okay, this axon ends in this kind of bulky projection, which is this bulb, and this is called the synaptic knob. Okay, that synaptic knob contains vesicles that and inside those vesicles what we have is a neurotransmitter that is called, in this case, acetylcholine. And the acetylcholine can stimulate the muscle fiber to contract. Now, in the other side, what we have the muscle fiber, right? So I'm gonna draw in here. I have here my muscle fiber, cylindrical, and right in here, when it's in close, not in close contact, but it's closely located or nearly to the synaptic knob, it kind of invaginates a little bit and folds, but don't worry, we don't need to know that structure. But the important thing is that the sarcolemma of this muscle fiber in there changes a little bit, and in here it has some receptors that can specifically interact only with acetylcholine. Okay, now the action potential, then it will travel from the neuron to the axon. When this action potential reaches, or this stimulatory wave reaches the synaptic knob, it stimulates the release of the neurotransmitter acetylcholine, by exocytosis. We already know that process, and it releases that neurotransmitter in this space between the synaptic knob and the sarcolemma of the muscle fiber. The sarcolemma on this area, this folded part of the sarcolemma, is called the motor end plate. The bulb or the knob is the synaptic knob, and the space between the synaptic knob and the motor end plate is called the synaptic cleft. So right here in the synaptic cleft is where the acetylcholine molecules are released, and then they can interact with their specific receptors. If there's other receptors in there, it won't interact. It needs to interact with the specific receptors. So the action potential now is initiated now in the skeletal muscle fiber, and will travel all the length of the sarcolemma, including the invaginations that we know as T-tubules. And in that way, we make sure that the action potential goes deeper into the cell and activates other or deeper myofibrils. Now, this action potential wave, what it does is that stimulate what they have on either side, which are the terminal cisternae. And the terminal cisternae gets all, you know, uh, blushed and releases calcium ions, which are all of these red dots that we can see in there. Now remember, this triad, the T-tubule and the two terminal cisternae, are located right on top of the overlapping area or zone in between the A and the I band. Right there, the calcium is released, so the calcium now can interact with the actin of the, let me show you here, can interact with the actin filament and to uncover the attachment site for the myosin, and the myosin can attach to the actin, pivoting or sliding the actin filament towards the middle, shortening the entire sarcomere and the entire muscle. Now, one thing important, the two factors in here. How do we stop this? Because if we keep, if this neuron keeps firing and we keep releasing in here acetylcholine, we will be in tetanus. This will be a sustained contraction, dangerous, and we need to control that. For that, we have right there in this space, in the synaptic cleft, there is an enzyme, acetylcholinesterase, that is in charge of breaking down the acetylcholine, so to shorten the duration of the muscle contraction. This is one important thing that I wanted to tell you. The other thing is that after that calcium is used by the actin filament, it goes back into the terminal cisternae for to be stored again. Now, the axon and the number of muscle fibers that the axon can activate or stimulate, we call that unit a motor unit. Let me grab this one better. Okay, a motor unit. Motor, kind of thick unit. And a motor unit is, again, an axon that branches and activates several muscle fibers, muscle cells, okay? The number of muscle fibers that can be activated by a single axon varies depending on the muscle and depending on the precision of the movements that we need to perform with that muscle. Examples: the motor unit of muscles of your thigh, for example, which movements or which contractions create movements that are not precise at all, can have or control like hundreds of muscle fibers. One axon controls or activate 100 or more of muscle fibers at the same time, okay? And all of those muscle fibers contract at the same time. The more delicate or the more precise the movement needs to be, the less muscle fibers we're going to include on that motor unit. So there's the motor units that control the muscles of the hands, where we need to perform precise movements, consist of one axon and maybe 3 or 4 muscle cells, muscle fibers. So this gives the ability of contracting just little parts of or little portions of every muscle, giving us the precision needed for certain movements. Now, what else? Let's see. describe this. That's it. Can I summarize all what we have described in this chapter? Step number 1. First, we receive the signal from the nervous system, and the nervous system sends a nerve to the muscle, and the nerve branches following the epimysium, perimysium, and endomysium until it reaches the muscle fiber. Once it reaches the muscle fiber, the action potential reaches the synaptic knob and releases the acetylcholine. The acetylcholine enters the synaptic cleft. There in the synaptic cleft, the acetylcholine can interact with the specific receptors located in the motor end plate, which is a modified part of the sarcolemma with several invaginations and blah, blah, blah. Now, the interaction of the acetylcholine with the specific receptors triggers or initiates the action potential now in the muscle fiber. And this action potential travels throughout the entire sarcolemma in the entire cell, including the invaginations that happen in the sarcolemma that forms the T-tubules. When the action potential reaches the T-tubule, it stimulates the terminal cisterna on either side. The terminal cisterna says, oh, and releases the calcium ions, and the calcium ions interact with the actin, and the actin releases or exposes the site where the myosin can attach. The myosin attaches in there, moves and pivots or slides the actin filament closer to the M line on the sarcomere and shortens the entire sarcomere. Review the changes that happens in the relaxed and contracted sarcomere. Now that we have contracted the sarcomere, we have contracted the muscle fiber. Since we contracted the muscle fiber and the endomysium of the muscle fiber is continuous with the epimysium and epimysium of the muscle fascicle and the muscle as an organ, all of these events are transmitted from the muscle fiber to the muscle fascicle to the entire muscle, and the entire muscle contracts, and then you can lift your dumbbell. Ready? One more video. Just talk about the types of skeletal fibers, and see you later, alligator.